all of our Friday evening lectures, a tradition that's been around since 1888 in some form or another, which continued on Zoom even during 2020, are made possible today by the generosity of donors and endowments that support the attendant events and the staff that make these possible. And tonight's lecture is the E.B. Wilson History and Philosophy of Science lecture. It's given annually on a topic relevant to the history and philosophy of science. And it was endowed by Richard Creeth and Jane Manshine, both longstanding and dedicated, generous members of the MBL community. Not just providing the resources to make this lecture possible, but the advice and the intellectual sort of interactions, advice, and guidance that help to make the history and philosophy of science a really integral part of the intellectual community here at the MBL. Edmund Beecher Wilson was a cytologist, geneticist, and one of MBL's earliest investigators and instructors. And he remained affiliated with the MBL as a researcher and as a trustee throughout his life. That's a theme here, actually, for today. We had our trustees meeting today, and people do grow attached to this institution for reasons that are obvious to many of you. But it touches people's careers, their lives, science, and society. And tonight, it's my pleasure to introduce one of our newest trustees, Susan Fitzpatrick. Susan will be introducing tonight's speaker. She's the president of the James S. McDonnell Foundation, who's been integral in supporting our history and philosophy of science activities in recent years, has been a committed member of this community, and we're very pleased to have her on our board as an advisor, supporter, and advocate. So please welcome Susan.
Thank you, Sebastian. And thank you all for being here. And many thanks to the people who make these very special evenings possible, especially, as you heard, our sponsors for this evening, Rick and Jane. Some time ago, I decided that I was going to join this movement to stop giving those kind of standard academic introductions that focus on detailing the many accomplishments and prizes that the speaker has been lauded with, because you can all Google that information. Instead, what I really like to do is explain why it's so exciting that we have Karl here to talk to us tonight. And even the briefest glimpse of Karl Matlin's CV demonstrates that he is a highly accomplished and feated scholar. In fact, if there are students in the audience, I would recommend that you study Karl's CV, because the keys to success in science are revealed. Go to the best places you can, learn from the smartest people you can, and work on the most important problems you can, especially problems that are important in advancing science in the service of society. But an eye-poppingly impressive CV is a necessary, but not by itself a sufficient condition to be a Friday night lecturer. These special evenings are reserved for those scholars who are also wonderful storytellers with a wonderful story to tell, and Karl is a perfect fit. In the preface to his 2022 book, Crossing the Boundaries of Life, which if you have not read it, I absolutely guarantee you it is a fabulous read. He describes the emergence of modern molecular cell biology, highlighting the research led by the Nobel laureate Günter Blobel. Karl, reflecting on his student days at Rockefeller University and his relationship with the Blobel Lab, writes, he, referring to Günter Blobel, considered the inner circle of his laboratory his family, and even though I was never a true member of his family, I was certainly an adopted child. I smiled when I read this. It reminded me that although Karl and I had not known one another until a few years ago, we met here in Woods Hole, we grew up scientifically in the same neighborhood. And anyone who has grown up in a city knows what that means. When you meet someone from the old neighborhood, there is an instant bond because of the shared experiences of people and places. So I was started as a graduate student in biochemistry at Cornell Medical New York Hospital, just as Karl was finishing up his PhD next door at Rockefeller University. I had close friends who are RU graduate students. I felt very drawn to that campus. One reason was its amazing history, much like the MBL. Another was that the Tower Building cafeteria had much better food than the hospital cafeteria. And famous scientists and Nobel laureates would gamely join a table of young students. The ethos of Rockefeller University was, to a young graduate student, was romantic ivory tower compared to the hustle of the New York hospital. And so I escaped to it whenever I could. It was an incredibly special scientific world populated by extraordinary scholars. It was magical. I offer this personal revelation only to spotlight why Karl's insights into the rise of modern molecular cell biology offer us a rare opportunity to hear a fascinating scientific story told by an internationally renowned scholar whose own experience with and contributions to that cell biology grant him a unique lens by which to bring life the research that defined a thrilling era of progress. Cell biology was reinvigorated by incorporating molecular biology in a way that embraced rather than reduced away the complexity. And in the 1970s, when this was really taking off, Karl was there, right in the midst of it all, and right at the right time. By being steeped in the Rockefeller environment and that amazing time and remarkable place, Karl adds to his life science and historical scholarship his deeply personal insights and how the work was accomplished and by whom. Science is at its core a deeply human activity. Driven by our curiosity, we cannot cease our questioning, regardless of whether we are gazing at the infinities of the heavens or peering inwards toward the intricacies of the cell. Tonight, we're about to explore the latter, and we could not have a more expert guide. Please welcome our speaker, Karl Matlin.
Thank you for that nice introduction. Today, we live in an unprecedented time in which it is easy to determine the structures of biological macromolecules, the proteins and nucleic acids that make up cells. But how do we know what these molecules do in cells? How do they contribute to making cells work? Take this protein molecule, for example. This representation is at a resolution of a few angstroms, 10 to the minus 10th meters, sufficient to see the path of the winding polypeptide chain, here portrayed as a ribbon. If you look closely, you might guess that the notations TM refer to parts of the polypeptides that are inserted into membranes. And you might wonder what the purple cylinder is and what the different colors mean. But what does this protein do? What biological phenomena does this detailed model by itself help us to explain? We can't tell. We have a molecule, but we have no molecular explanation. Later, I will return to this molecule, but for now, let's move to an earlier period. Edmund Beecher Wilson, who you were just introduced to, and for whom this lecture is named, spent most of his summers here at the MBL and was famous not only for his research, but also for a textbook that he wrote that went through 3 editions. In the 1925 edition of this textbook, called The Cell in Development and Heredity, he stated, "Whether structure or function is the primary determining factor in vital phenomena is a question that has been the subject of debate for many generations of biological philosophers. As thus stated, however, the question has proven barren, for all students of the problem have, in the end, had to admit that structure and function are inseparable. Thus has arisen a dilemma which belongs to the fundamental philosophy of biology and may be set aside here as practically insoluble. Wilson viewed the cell, as depicted in the schematic drawing from his book, as a membrane surrounding protoplasm, or as we now say, cytoplasm, containing also a nucleus, and a variety of permanent or temporary inclusions, what we now call organelles. The problem or dilemma, to paraphrase Wilson, was that all living cells, and by extension, all living organisms, require such an organized structure to be alive. The functions of living cells, what Wilson called vital protoplasmic activities, are, however, carried out by chemical reactions that occur within this structure, but cannot be appreciated from the structure alone. This particular dilemma had a practical side that at the time of Wilson's statement was an obstacle to the study of cells and blocked explanations of how cells work chemically. Cytologists realized that to access and understand the chemical reactions in living cells, that is, to decipher vital processes at the molecular level, it was necessary to disrupt and kill the cells. However, cytologists had no confidence that the reactions uncovered by studying dead cells would provide any insight into the chemical reactions that were important in living cells. In Wilson's terms, if organized structure is essential to life, how can you learn anything about the chemistry of life by destroying the structure? Such concerns were frequently voiced by biologists in the late 19th and early 20th centuries. Quoting a Swiss botanist, the German cytologist Oskar Hertwig remarked in 1895 that protoplasm cannot be placed under different conditions without ceasing to be protoplasm, for its essential properties in which life manifests itself depend on a fixed organization. As a statue ceases to be a statue if broken up into small pieces of marble, so a body of protoplasm is no longer protoplasm after the organization, which constitutes its life, has been destroyed. We only examine the considerably altered ruins of the protoplasm when we treat the dead cells with chemical reagents. Similarly, in 1899, the physiologist Max Verworn stated that living substance must be killed before its chemical composition can be learned. Paradoxical as this may sound at present, it is the only way by which knowledge of the chemistry of living substance can be obtained only by deductions from chemical discoveries in the dead object. Deductions, the correctness of which can be proved experimentally in the living object only in rare cases. This alone is responsible for the excessively slow advance of the knowledge of the chemistry of vital processes. Today, I want to tell you the story of how this dilemma was overcome through the development of modern cell biology. There is a distinct history. Advances did not occur simply because of new instruments, although these were important, but rather through the concerted efforts of a group of biologists over an extended period of time. The key development was the creation of a research strategy that made sure that the chemical functions of the cell were always studied in the context of the cellular structure. This permitted cellular phenomena to be explained at the molecular level. The first steps were taken, of course, at the University of Chicago. Robert Bensley, a noted anatomist and histologist, shown here in an image taken at the MBL, and his collaborator, Norman Hoare, wanted to study the distribution of chemical activities in cells by staining microscopic sections of tissue. This approach, however, was beset with many problems, some related to the concerns voiced by Hertwig and Verworn. As Bensley stated, the investigation of the distribution of chemical substances in cytoplasm has been beset with extraordinary difficulties. The excitation of cytoplasm by even minute quantities of reagents may so disturb the equilibrium between several phases of cytoplasmic structure that the distribution of substances in the cell as a whole is completely changed. As an alternative, they tried freeze-drying the tissue, cutting it into thin sections and extracting material from the sections for chemical analysis while monitoring the extraction process in the microscope. This strategy was also limited, but Hoare noticed that the procedure released mitochondria, an organelle that they were interested in, from the sections. Based upon this, they decided to crush the tissue, extract it, and then separate the mitochondria from other parts of the tissue by spinning the disrupted tissue in a centrifuge. With this approach, they began to make progress. Centrifuges like the one that Bensley and later others used are instruments designed to separate small particles of material, such as the fragments of broken cells, using centrifugal force. Samples are placed in tubes that are inserted into rotors, such as this shown here, this silver object. The rotor then spins at high speed, such that the fragments, larger fragments go to the bottom of the tubes, while smaller and intermediate-sized fragments are distributed along the length of the tube. Over time, such instruments became very sophisticated, spinning at speeds of more than 50,000 RPMs in a refrigerated vacuum chamber. Bensley, who was near the end of his career when he did this work, did not pursue his new strategy for long. Almost immediately, however, another scientist, Albert Claude, shown here, at the Rockefeller Institute for Medical Research, stumbled upon a similar approach. Claude was trained as a physician in Belgium and moved to New York in 1929 to conduct cancer research, an interest motivated by his mother's death from breast cancer. Claude's background was rather unusual. During World War I, he had been a British spy, even though he was very young. After the war, he was permitted to enter medical school with no high school education as a reward for his service. Significantly, as we will see later, Claude was not trained as a cytologist and was likely unaware of the dilemma about breaking open cells that cytologists worried about. In New York, Claude adjusted well, and in addition to his work in the lab, circulated socially in the New York arts community. He even took a microscope and specimens to the new Rockefeller Center so that Diego Rivera, shown here, could incorporate the microscope and images of cells into his ultimately doomed fresco called Man at the Crossroads, and you can see some of this here. As Rivera stated, the microscope makes visible and comprehensible to man infinitesimal living organisms connecting atoms and cells with the astral system. Rivera's work was soon destroyed because somehow the Rockefeller family didn't like the fact that he incorporated an image of Lenin into his mural. In the lab, Claude was tasked with isolating a transforming principle that Peyton Rouse had discovered transmitted cancer in chickens. Some in the group believed it to be a virus, while others considered it likely to be an enzyme, leading Claude to try and purify it from disrupted cells using biochemical procedures. He eventually heard that a British research group was having success separating the factor from tissue extracts using a centrifuge, and he decided to try this approach himself. When he did, he discovered that he could resolve the cancerous cell extracts into several fractions containing different sized particles, some of which he believed to be the transforming material. The problem was that his control, an extract from non-cancerous tissue, had the same distribution of particles. Claude soon realized that he devised a method to separate and purify different parts of the cell called cell fractionation. Claude was a very careful worker, separating multiple fractions by centrifuging his cell preparations at different speeds and for different times. As you can see in this flow chart, he would start with an extract, centrifuge it, separate it into a large granule fraction at the bottom of the tubes and a supernatant at the top, and then further centrifuge the supernatant at higher speed until he resolved a small granule fraction. Claude's approach had two essential features. First, as he fractionated cells, he accounted quantitatively for every part, constructing a balance sheet similar to the one shown here. This allowed him to eventually associate particular biochemical reactions with particular parts of the cell, an insight into the functions of those parts. And second, at every step from the intact tissue to the separated particulate fractions, Claude monitored his progress microscopically. As he later said, in the presentation of the results, it would be difficult to separate the biochemical work from the morphological observations, since a microscope has constantly served as a guide or check for the chemical and biochemical studies. In essence, Claude's use of the microscope during cell fractionation enabled him to link his cell fractions to the intact cell, preserving the cellular context during his biochemical analysis. This was a step toward resolving the cytologist's dilemma, because the structure of the cell was not obliterated, but rather carefully disassembled. Even though Claude's use of the light microscope was essential, it was also limited by its inherent resolution that he and others suspected left regions of the cell what they called optically empty, and the cellular components that they suspected were there invisible. In the early 1940s, a young scientist, Keith Porter, shown here, later an MBL director and who had a lab just upstairs from here, joined Claude's laboratory and began collaborating on a new project using an electron microscope to look at cells. Electron microscopes had been developed in Germany in the 1930s, but their use on biological specimens had been limited. Even though electron microscopes could magnify objects thousands of times more than light microscopes, the environment inside the electron microscope was so harsh that biological specimens needed to be specially preserved and prepared. They also had to be very thin, even though the electron beam that substituted for light in the electron microscope was powerful. It could not even penetrate something as thick as the nucleus in a single cell. Porter came up with the ingenious solution of growing cells in culture on a sheet of plastic, such that they spread out very thinly. He then fixed the cells with preservatives, cut them out of the plastic support, and inserted them into the electron microscope. This is the first composite electron micrograph of a eukaryotic cell. What he and Claude saw was astounding. In one optically empty part of the cell, there was now a complicated structure never before seen that they described as a lace-like reticulum. In 1949, Claude returned to Europe, leaving Porter behind. In the meantime, the techniques of both cell fractionation and electron microscopy improved at Rockefeller and elsewhere, making new discoveries possible. Even before Claude left, Porter had already been joined by a new colleague, George Pallotti, shown here. Pallotti was an MD and anatomy instructor in Romania who had become more interested in research than medical practice. When post-war conditions made it impossible to work in Romania, his scientific mentor, who frequented the MBL in earlier years, arranged for him to join Robert Chambers' lab at NYU in 1945. Impossibly Pallotti's first summer in the United States, he joined Chambers here at the MBL, where he did experiments on sea urchins. Back in New York, he saw Claude's electron micrographs, probably in a seminar, and was so impressed that he moved to Rockefeller from NYU to work with Claude and Porter. At Rockefeller, Pallotti learned cell fractionation from Claude and electron microscopy from Porter. When Claude returned to Europe, Porter and Pallotti became de facto heads of what had become electron microscopy service at Rockefeller, which eventually matured into the Laboratory of Cell Biology. In collaboration with Porter, Pallotti began the morphological characterization of the lace-like reticulum, now renamed the endoplasmic reticulum, abbreviated ER. And by the early 1950s, Pallotti took over the project himself. Using electron microscopy, he established that the endoplasmic reticulum, seen here in one of his micrographs, was present in all eukaryotic cells and resembled a series of anastomosing membrane tubules. He also discovered what he called a particular component of the cytoplasm that often appeared to be bound to the endoplasmic reticulum membrane, which shows up in this early micrograph as these black granules. These particles were labeled, for a while they were called pallotisomes, and later they were renamed ribosomes and were linked in biochemical studies by others to the synthesis of proteins. Inspired by Claude's use of microscopy as a guide in cell fractionation, Pallotti began to develop what he called his own integrated strategy, which linked morphological characterization of cells and cell fractions with the electron microscope to biochemical analysis of these fractions. Pallotti was particularly interested in the process of secretion, the synthesis and transport of proteins out of the cell. To investigate this, he recruited the biochemist Philip Siekowitz, who had earlier worked on the biochemistry of protein synthesis. In a series of studies in the late 1950s and early 1960s using pancreas exocrine cells, cells that secrete digestive enzymes into the intestine, Siekowitz and Pallotti demonstrated that the endoplasmic reticulum of the rough variety, as he always said, that is the endoplasmic reticulum with bound ribosomes, broke apart during cell fractionation into small closed membrane vesicles with ribosomes still bound to the outside of the membrane, which you can see in this early micrograph. They called these vesicles rough microsomes. They also showed that the synthesis of secretory proteins begins on the bound ribosomes and that these proteins somehow cross the ER membrane to enter the pathway leading out of the cell. Let's have a look at his integrated strategy in more detail. This was a carefully orchestrated approach that identified a cellular phenomena and then used microscopy, cell fractionation, and biochemical analysis to localize particular events to specific parts of the cell. Claude began studies by examining electron micrographs of intact cells, such as this one, and then using these images and other existing information to propose a hypothesis about the cellular phenomena of secretion. This hypothesis suggested that the synthesis and transport of secretory proteins occurred in vesicles. Claude then systematically broke the cell apart and identified and localized the parts, the organelles, morphologically by comparison with the intact cell. Now he could proceed with biochemical analysis of the characterized parts and conduct experiments on the parts to test his hypothesis. When he did this, he was now confident that the cell parts he was working on accurately represented the same parts in the intact cell. In this way, Claude addressed some of the earlier concerns of the cytologists who worried about working on disruptive cells. Through subsequent iterations of this strategy, going around this cycle, he gathered more and more detailed information about the phenomena under investigation, secretion. This process is what's termed a recursive process because each iteration builds upon the results of the last one. At this point, Claude was the sole leader of the Laboratory of Cell Biology after Porter left in 1960 for Harvard. By the early 1970s, he had established with Siekowitz and other collaborators in the lab that secretory proteins in the pancreas first cross into the ER after their synthesis, are transported through the cytoplasm and eventually out of the cell in vesicular carriers. While this accomplishment was enough to win Claude with Albert Claude and Christian Dedeuve a Nobel Prize, in fact, Claude had not identified the molecules in the cell that carry out the secretory mechanism. That is, he had not explained secretion at the molecular level. This would require a further development of his integrated strategy, one that pushed it to the molecular level. This next step would be taken by a new person in the laboratory, Günter Blobel, shown here in a picture from 1982. Blobel had arrived in the lab after completing a PhD in biochemistry at the University of Wisconsin, studying the circulation of ribosomes between the free and bound states. Prior to moving to the United States, Blobel had completed an MD in West Germany after migrating from East Germany in the 1950s, before the Berlin Wall made such moves impossible. When Blobel got to Rockefeller, he had at first drifted around, not being sure what he was going to work on, until he began a collaboration with one of Claude's protégés, David Sabatini. Sabatini had already shown that ribosomes may, in part, stick to the ER membrane by what's known as the nascent chain. That is, the new secretory protein being synthesized as it grows out of the ribosome. He and Blobel began experiments to figure out exactly how this worked. In 1971, Blobel presented a speculative model created with Sabatini at a meeting. The model suggested that the first part of the protein synthesized, here marked with an X and labeled the targeting sequence, directed or targeted secretory proteins during their synthesis to the surface of the ER membrane, and then, in some vague way, facilitated their movement across the membrane into the inside, shown here. About a year later, work by others at Cambridge University, which started out as not initially focused at all on the process of protein secretion, provided, in fact, the first evidence that the model proposed by Blobel and Sabatini might be true. At Rockefeller, Blobel was energized by these results and set about repeating and extending them. By this time, however, Pallotti had departed for Yale and Sabatini for NYU. Blobel was on his own with the project. He first created a completely cell-free or in vitro experimental system to pick apart the mechanism. His system consisted of microsomes, mRNA, a complex mixture of enzymes and metabolites that would help catalyze the synthesis of secretory proteins, and radioactive amino acids to enable him to detect the newly synthesized proteins. When these components were incubated together, Blobel hoped he could demonstrate not only that a secretory protein had been synthesized, but also that it had crossed the microsomal membrane into the interior. He hoped to do this by adding proteases to the outside of the vesicles, such that any proteins that cross the membrane would be protected by the membrane and not degraded. Of all the components, while proteins have been synthesized using similar systems many times, the key component that Blobel added was the microsomal vesicle. Microsomes represented an actual part of the cell that had been, through the work of Pallotti and others, linked directly to the intact ER, to microsomes, and eventually to entering Blobel's system. The microsomes were the cell surrogate, ensuring that the molecular event that Blobel studied occurred in a cellular context. In his first experiments, he compared the synthesis of proteins in the cell-free system in the presence of microsomal membranes or in the absence of microsomal membranes. When he examined the sizes of the synthesized proteins using a technique called gel electrophoresis, sort of illustrated here, in which proteins mixtures are loaded on the top and then electric current is applied so they migrate through this gel, and autoradiography to detect the radioactive proteins. He observed that proteins synthesized in the presence of membranes were slightly smaller than those synthesized in the absence of membranes. Also, using proteases, as I described before, he demonstrated that the smaller proteins had entered the inside of the microsomal vesicle because they were protected from degradation, while the larger proteins synthesized without microsomes were completely degraded by proteases. When he sequenced these two sizes of proteins, he discovered that the larger ones had a short extension of amino acids at the beginning of the protein that was apparently removed as the proteins passed through the microsomal membrane. Based on these experiments, Blobel proposed a more sophisticated version of his original model that he called the signal hypothesis. According to this proposal, which is illustrated here, The synthesis of secretory proteins begins with the short extension which he calls the signal or signal sequence. When the signal is exposed at the surface of the ribosome, it causes the synthesizing ribosome messenger RNA complex to bind to the ER, or microsome membrane, in his experiment, leading to the assembly he proposed of a tunnel or channel through the membrane through which the new protein would move to the interior of the ER, or microsome. During its passage, the signal sequence would be cut off with the new protein. Over the next 20 years, Blobel's lab and others, inspired by his accomplishments and using his cell-free system, showed that the signal hypothesis operated not only in mammalian secretory cells, but also in diverse array of organisms and tissues, including organs, parts of fish, baker's yeast, and bacteria. Most importantly, Blobel's experimental system incorporating a part of the cell, the microsome, as a surrogate for the ER in the living cell, permitted the actual molecules carrying out the mechanism of protein targeting and transport to and through the microsomal membrane to be identified, isolated, and characterized. These included something called the signal recognition particle, or SRP, and its receptor, which bound to the signal sequence coming out of the ribosome and directed the whole synthesizing complex to the membrane, the channel or tunnel protein called Sec61, and also signal peptidase, the protease responsible for cutting off the signal sequence. I've argued here that the development of the integrated strategy of Claude and Pallotti, which combined microscopy with the systematic fractionation or decomposition of the cell, and carried to the molecular level by Blobel, began to resolve the apparent antithesis between structure and function noted by E.B. Wilson. It also addressed the cytologist's practical dilemma, the fear that disruption of cells would uncover biochemical reactions that are artifacts, not representative of a living cell. What's the basis of my argument? The integrated strategy begins with an appreciation of the intact cell, in these cases as represented in electron micrographs, a holistic perspective. Then, in a series of iterations of that strategy, various parts of the cell are isolated and identified, and their structure in isolation is compared with the structures in the intact cell. Then, these parts, still linked to the intact cell, are systematically broken down into their molecular parts using an experimental system that retains not only essential aspects of cell structure in the form of microsomal vesicles that resemble the ER in this case, but also essential aspects of cell function, the ability to transport proteins into the vesicles. In this manner, the cell biologists conducting these molecular investigations in a cell-free system had greater confidence that the molecular mechanisms that they discover accurately represent what goes on in the living cell. In the years after the events I described here, the integrated strategy of Claude, Pallotti, and Blobel continued to be used, and I believe is still used today. In fact, I think it's common. Cell-free systems containing parts of the cell acting as cell surrogates led to discoveries into molecular mechanisms of membrane trafficking through the Golgi complex, endocytosis, nuclear transport, and many other phenomena. Even today, when advanced light microscopy with fluorescent probes has replaced electron microscopy and cell fractionation, I believe that essential aspects of the strategy are still applied and are still yielding molecular explanations of biological phenomena. But that's for another day to discuss. And what of that protein that I showed you at the beginning of my talk? Well, now we know, through the research strategy of cell biology, that this protein is in fact the channel that guides proteins through the endoplasmic reticulum membrane. Here is the original structure I showed you. Here is a rotated version with a membrane colored in, and here are cartoons of how it functions. Now, we have both a molecule and a molecular explanation of a biological phenomenon. And finally, thanks to these organizations for support for the work that led up to this. Thank you.
Questions? Please do wait for the microphone, and we may have some online as well. Yep, I'll come back.
Hi, thank you for coming. Just out of curiosity, what do you think the most important cellular biology discovery within the past 10 years has been?
I don't know if I want to answer that. Well, I think I have to say, I won't name a specific discovery, but I will say that the developments of advanced light microscopy with fluorescent probes has allowed the dynamics of cellular events to be examined. I think that the approach is very similar to this, but I think that that's the biggest advance. I can't really put my finger on what's the best thing in the last 10 years. And it probably wouldn't be a good idea to say that.
Thank you for the lecture. So I was wondering, what was the real barrier for the cytologist at the beginning? Was it a conceptual barrier or a technical one? Because I think that the notion that you can actually take an entity, decompose it into parts, study the parts in a recursive way, I think that that's something that in biology has been done before, anatomy and all of that. So to me, it would seem like it's more the technology that really enabled this. These advances, but I think that you were arguing also there was a conceptual leap somewhere, so I'm just trying to...
Well, I think it was both a conceptual and a technical problem. You have, and I've limited my discussion to cytologists, because you have to realize that in parallel was development of biochemistry. Now, biochemistry didn't care about structure. As far as they were concerned, you could obliterate the cell as long as the enzyme activities that they were interested in were still going. Cytologists knew that this type of biochemistry was possible, but they had spent their lives looking at the form of the cell and imaging the cell. Their primary, up until, let's say the second third of the 20th century, their primary tool for investigation was a microscope. And they were just very, very hesitant to destroy the structure. There's even, not to promote my book, but in my book I talk about something that Keith Porter said after Albert Claude died. And even that late, which would have been in the 1940s and almost 1950, Porter said that Claude had a tremendously hard time from some people because they said, how can you possibly destroy such a beautiful structure? And these attitudes went on for a really long time.
Hi, thanks so much for your talk. I think maybe the answer to my question might way in the back here.
Oh, okay.
Sort of might relate to that intersection of biochemistry and cytology. And I was wondering if in all of your research you've come across some of the serendipity of choices that were made to accomplish these cell-free assays, and in terms of cell type or the species, or
What was the last?
The question is, do you have some fun anecdotes to us about sort of the serendipity of choosing a certain cell type or from a certain species or a certain organism to set out to do these assays? If this wasn't all done with plant cells and it wasn't all done with bacterial cells and so on, and I wondered if, what you thought about how the choice of cell model was important for making these discoveries.
Well, in the case that I talked about, particularly regarding Pallotti at Rockefeller, it was not serendipity. What I can tell you, I always thought, even when I was there as a student, years later after this, what was unusual about Rockefeller was that the focus was on a phenomena, a general phenomena that they thought was a biological phenomena of interest. And then they would go looking for a system that they thought was amenable to study, but also exhibited this, the things that, the phenomena in a big way that they wanted to look at. So for example, Pallotti and Sikovits started out working on liver. And they made some progress in characterizing the function of the endoplasmic reticulum in the liver. But at a certain point, they moved to the pancreas because it was a professional secretory organ. But they had no qualms about jumping around. One of their postdocs a little later, when they were trying to establish an in vitro system, used pigeon pancreas because it was low in ribonuclease. In the same department, they wanted to work on membranes, so they started working on Chlamydomonas and algae. So they had the general attitude that if they were looking at a biological phenomena and trying to establish a mechanism, that mechanism would be fundamental and that it would be replicated to some degree in whatever living system they might look at. And Blobel's work definitely established that when they found the same, proteins, secretory translocation proteins and things in all kinds of organisms.
Thanks, Karl. I mean, you described experiments by many of the greats of cell biology, and the electron microscope was such a revolution. And it's interesting to me to see how they were carefully fractionating parts of the cell and then doing biochemical assays to correlate the biochemistry with what they saw functionally. But still, there were many skeptics, who thought that they'd just broken the cells and things weren't working. And so I'd just like to hear more about, the skepticism, and what won out. How did these, how did the careful investigators win out in the end? Because that seems, was it just their care? Did it take a long time for other people to repeat their experiments? What were the key factors that really, got them the acceptance that they were right?
Well, I'd say in terms of controversies, there definitely were controversies about the pathway that Pallotti established. I just mentioned, it was always ironic to me that when Pallotti got the Nobel Prize in 1974, I think it was 74 and it was awarded in 75, something like that. At that time, they published his Nobel lecture in Science. The following issue of Science had a review from some guy in California that claimed it was all wrong. And it was published in Science. One of the most important things, which I mentioned here, and I just really have to emphasize, was this use of a balance sheet when they did fractionation. I mean, tons of people after that fractionated cells, Bensley fractionated cells, but the way they would do it, and unfortunately the way a lot of people do it now, is break open the cells and isolate what you think you're interested in and throw the rest away. They accounted for everything. So if there was an enzyme, a particular enzyme, they looked at where that enzyme was concentrated in the cell, which fraction. And they tried to recover as much a percentage of it in all the fractions together. So I think that was one factor in the acceptance of the work that they did. So I don't know if I've answered your question, but.
We have a question online, and then we'll have one more in the audience.
Where are we headed? Will the molecular level be replaced by a level showing the organelles that transport individual amino acids into the ER in the correct sequences for protein synthesis?
Can you repeat that? I didn't quite understand.
Yep. Where are we headed? Will the molecular level be replaced by a level showing the organelles that transport individual amino acids into the ER in the correct sequences for protein synthesis?
Are you talking now about synthetic biology? Or are you talking about a resolution of what is occurring?
I unfortunately lack the context from the online questioner, so.
Oh, okay.
If you can't answer it, that's fine.
They may clarify while we take the one in the audience.
Okay, thank you. There's a whole range of intracellular components, and of course your presentation focused on one of those. I was just curious, has there been an analog history relating to what is arguably the most important component of a eukaryotic cell, and that is the nucleus, in terms of explaining the biochemistry of events in that area of our cells?
So what is your question exactly? I'm sorry, I have a little bit trouble here.
Okay, just to recap, your focus has been on one component of eukaryotic cells, and of course the ribosome, the rough endoplasmic reticulum, secretory proteins, etc. Is there an analog history that relates to the nucleus, which is arguably the most important component of a eukaryotic cell, and how its biochemical events were ascertained?
Well, that's a very interesting question, because I think the history of that is being written. For most of my career, the events of the nucleus, in particular gene expression, regulation of gene expression, were treated as a one-dimensional problem, essentially. So there were certain factors which bound to DNA and either turned on genes, suppressed genes, synthesized genes. So that was considered how things worked. In parallel, since the 1970s, let's say, and on, there was a cell biology of the nucleus. In fact, Blobel's other major project in his lab for many years was the nucleus. And there's been established a huge amount of information and structure about transport in and out of the nucleus, how it's regulated, how material moves in and out through pores, and so on. But fairly recently, individuals working on gene expression have realized that the regulation of gene expression is actually a three-dimensional problem, and that chromatin is arrayed in the nucleus in a particular orientation, morphology, in 3 dimensions, and that changes with gene expression. So suddenly, the idea that regulation of gene expression is a, let's say, molecular biology problem with dealing with the sequence of DNA and the binding affinities of certain proteins, now it's really a cell biology problem in the sense of the form of the cell, the form of the nucleus, and the regulation of the gene expression by changes in that form and the form of chromatin. I don't know if that answers your question, but.
I came dangerously close to commenting on the first question, so, which we can pick up at the discussion at the reception following on the back deck of Swope. So please thank Karl one more time.